As we have seen in the process organism, step by step, that first step was isolation, then is pure culturing, and then is screening, and then is identification. But in this whole process of the process organism is very tedious and time taking. But after the identification, then the problem and the main task for the process organism is the storage and the preservation. And the storage and preservation in such a way that the maintenance of that culture. If we just isolate and then we have not a proper storage and preservation of that culture and isolate, then that organism can lose its potential for the production of our desired product. So the storage and the preservation is very critical in the fermentation process for the continuously having the same quality of the product. So there are the isolation of a suitable organism for commercial process may be long and very expensive, as I have already told, and then there should be a desirable characteristics. Also, the culture used to initiate an industrial fermentation must be viable and free from contaminations. Because if we have an isolate and having a pure culture and then we identify and we have not good storage and preservation, then there is a chance of contamination. So the industrial culture must be stored in such a way to eliminate the genetic change, just to have the proper maintenance, so to protect against the contamination and retain viability. So three major objects. First one, having genetic stability, and then no contamination and retain viability. So these major three objects that can only be met through the proper storage and the preservation of that culture. So the organism may be kept viable for repeated subcultures into fresh medium, but at each cell division there is a small probability of mutation occurring, and because of the repeated subcultures involves very many subdivisions, there is a high probability that the strain degeneration would occur. Because when there is a mitosis or a meiosis occur, then there is a crossing over. In result of the crossing over, there is a mutation, which we called as spontaneous mutations. So in a result of those mutations, natural mutations, in result of that, there is a chance of having the lose of that potential. So also the repeated subcultures carries with it the risk of contamination. Thus, the preservation techniques have been developed to maintain that culture in a state of suspended animation by storing either at reduced temperature or in dehydrated forms. So as concerned the storage at reduced temperatures, so first choice is storage on agar slant. We just store those agar slant on which the growth has already occurred at in the refrigerator at 5 degrees Celsius or a freezer at minus 20 degrees Celsius and subcultured at approximately 6 monthly intervals. The time of subculture may be extended to 1 year if the slope are the covered with sterile medicinal grade mineral oil. So that concerns the next, the storage under liquid nitrogen. And we know that what is the temperature of the liquid nitrogen, that range from the 150 to minus 150 degrees Celsius to minus 196 degrees Celsius. So the metabolic activities of microorganism may be reduced considerably by this temperature, so which may be achieved using only the liquid nitrogen refrigerators. Fungi, bacteriophage, viruses, algae, yeast, animals and plant cells and tissue culture can all be successfully preserved by using these methods. So techniques involve growing a culture to a maximum stationary phase, resuspending the cells in a cryoprotective agent such as 10% glycerol and freezing the suspension in a sealed ampule before storage under the liquid nitrogen. So glycerol is mostly used non-solidifying agent, which we called as cryoprotective agent. So as concerned, then the second choice, that is the storage in dehydrated form. First one is a dried culture. So the dried soil cultures have been used widely for culture preservation, particularly for sporulating the mycelial organisms. Moist, sterile soil may be inoculated with the culture and incubated for several days for some growth to occur, and then allowed to dry at room temperature for approximately 2 weeks. So in this dry method, first we put some soil into that medium, and then that have to dry for 2 weeks, and then that dry soil may be stored in a dry atmosphere or preferably in a refrigerator for the storage. The technique has been used extensively for the storage of fungi and actinomycetes, as reported by this. But he observed that the 1800 actinomycetes dried on soil, about 15% were viable after 20 years of storage. So it is a very conventional method used for the storage of the cultures. As concerned, the next is storage in dehydrated forms, that is the lyophilization. So during the lyophilization, which we called as the freeze drying, a suspension of microbe is quickly frozen at temperature ranging from -54 to -72 degrees Celsius, and then the water is removed by high vacuum, which we called as sublimation. While under the vacuum, the container is sealed by melting the glass and a high temperature torch. The remaining powder, like the residue that contain the survivors microbes, can be stored for years. So most of the organisms that we collect from the different culture banks, we have a lyophilized form of those microorganisms. So the lyophilization is a very convenient for the surface cultures collection, because once dried, the culture need no further attention and the storage equipment like refrigerators. So this one is a cheap and a reliable method of the storage of any process organism.